Hi everyone, and welcome to part 2 of the blood chapter. So we last time left off talking about different types of anemia. So now we're going to move on and talk about blood typing. Okay, so this can sometimes be a confusing topic for students, and so I like to kind of break it down into this chart that we're going to fill out. And I think if you practice this over and over and over and try like making this chart yourself and filling it out without looking at any notes to kind of make sure you have a good understanding of it, that you'll be okay. It's the more you practice it, the easier and more logical it becomes. But when you first go through, if this is your first time hearing about blood typing, and we'll talk about transfusions and possible transfusion reactions, if this is your very first time going over that material, it's gonna seem like a lot. So just keep practicing it. Okay, so that being said, a little disclaimer. Let's go ahead and start kind of defining some terms, and then we can talk about blood types. Okay, so our first term is an antigen. An antigen refers to anything that can cause an immune response. All right, so anything that causes an immune response. And so with our red blood cells, when we talk about these, all right, not writing red blood cells. Yeah, okay, response, sorry. All right, so with our red blood cells, when we talk about them, what gives them that blood type of, A positive, B negative, AB, that kind of thing, is the antigens that are on the surface of those blood cells. Okay, and so we'll talk about those. All right, and so our next term is an antibody. Okay, so an antibody is what your body produces as the immune response to an antigen. Okay, so this is the immune response, and then typically antibodies are drawn as kind of like a Y shape. Okay, like this. All right, so that would be an antibody. And its job, what it's going to do, is bind to an antigen. All right, and so it will then, by binding to it, cause the antigen to clump. And so that is referred to as agglutination. Okay, so agglutination is clumping of an antigen, all right, due to the binding of that antibody. All right, so due to antibody binding. All right, and so the reason that when antibodies bind to antigens, it causes them to clump is that every antibody can bind to different antigens. Okay, so the kind of ends of the Y here, each end can bind to an antigen. All right, and so you'll have little clusters, and then the antibodies can kind of link together. So let's say if we have like an antigen bound here to this one and another one bound here on this antibody, and then you can have another antibody bound to this one, all right, with an antigen here, and then another antibody, and so on and so on. And so they form these big clumps. That's agglutination. All right, so now let's talk about blood typing and how these terms relate to that. Okay, so blood typing, we use kind of two different groups. So we use the ABO system, and we use the plus-minus system. All right, and so with these, they are based off of what antigens are present on the cells. Okay, so blood typing is the antigens on the surface of the red blood cells. All right, so hopefully in A&P1 you learned about some membrane proteins, and one of those membrane proteins you should have learned about was a glycoprotein. So just a protein embedded in the membrane that has a bunch of sugars on it. That is what these antigens on red blood cells are. All right, so they're just membrane proteins with some sugars, and we identify them using the letters A, B, O, and RH. Okay, and so blood types are genetic also. So this is coded for by the DNA, and it's passed down from your parents. Okay, so there are actually 30 different types, like groups and way to, blood types. So there are 30 different possible ways to take somebody's blood type out there. The ones that we use are these ABO and RH antigens, and that's because they are responsible for the most severe transfusion reactions when you're, giving someone blood and that sort of thing. Okay, so these are the ones we focus on. All right, so with the ABO antigens, all right, the way they work is the patient is type A if they have the A antigen on their blood cell. They're type B if they have the B antigen on their blood cell. And they're type O if they don't have either an A or a B, okay? So O is kind of like, think of it as like 0. O as 0, meaning they don't have any of those antigens. RH is going to be, so we'll just put, we have the possible ones here are A, B, and they can have both A and B antigens or O. Okay, and then with RH, we have either positive or negative. So the individual is positive if they have that RH antigen, and they're negative if they don't have it. Okay? All right, so let's go through and fill out this chart first for the antigens, because that's what we just talked about. So let's only focus on that middle column for right now. Okay, so I'm going to zoom in a little just so we can see. All right. So for an A negative blood type, the antigens on the surface of the red blood cell would just be A. All right, so whoops, I meant to use this. There we go, would just be A, okay, because it's A negative, so that means they have the A antigen and no RH antigen because it's negative. All right, so then with A positive, they would have the A antigen, and then they would also have an RH antigen because they're positive, right? So they're positive, meaning they have that RH antigen. Okay, and so then B would only have a B antigen. It's negative, so no RH. B positive would have a B antigen and an RH antigen because it is positive. All right, hopefully you're kind of getting the hang of this. This part is pretty straightforward. So then A, B, whoops, that should be a B. All right, and it's negative, so there's no RH. And this one is A, B, and it's positive, so there is an RH. Then last is our type O's, okay? So O means there's not an A or a B. And so then it's negative, so they don't have an RH either. So this would be none. So there would be no antigens at all on the surface of these red blood cells. All right, so none of the ones that we're looking at. All right, and then this one, we wouldn't have A or B, but it's positive, so there would be an RH antigen just by itself. Okay, so those are the antigens on the surface of the red blood cells for the individuals with these blood types. Okay, so all of these blood types, these are all the possible ones, and the antigens that are on their surface. Now, what we are going to do is we are going to fill in the next side of this chart, and for this, we are looking at what antibodies, and specifically what antibodies are made by an individual with each blood type. All right, so with that specific blood type. And so what is really important to remember here is that you make antibodies against the antigens you do not have. Okay, so I'm going to write that just so you remember it. All right, so antibodies are made against antigens you don't have. All right, so antigens that you do not have. Okay, because obviously you don't want to make antigens again or antibodies against your own antigens. That would cause your blood to clump to a glutenate. Okay, so you want to make antibodies against the antigens your red blood cells don't have. That way they'll be against anything that is foreign. All right, so let's start with our our A negative here at the top. Okay, so we can see the antigen that they do have is A. What's missing of all our, so we have 3 possible antigens, A, B, and RH. What is missing is B. All right, so we're going to have an antibody against B, and we're missing RH. Okay, and so RH is a little unique compared to A and B. All right, so we are going to say RH if exposed. Okay, and so that means, so this person, I'm going to put this kind of below, it's getting a little crowded. All right, so RH if exposed. Okay, so that means that the way it works with the RH antigen is that a person will only have antibodies with it if their blood, say they were given a transfusion, and that transfusion, this individual was given a transfusion, and that blood they were given contains like an RH like this. So they were exposed to that. Their body prior to that didn't have antibodies against it, but now that it's seen that, you know, weird RH that's not normal in the body, it started making antibodies. Okay, and so now they will have the antibodies. So it's RH if exposed, because first their immune system has to see the foreign RH in order to make those antibodies. Okay, so that's a distinction that's only important with our RH. Okay, all right, so A positive, the only antigen missing is B, so they would only make antibodies against B. All right, B negative, so we're missing an A antigen and an RH antigen. So they would have antibodies against A, and they would have antibodies against RH. And again, we'll just do E here, right? That would be if exposed. Okay, so RH if exposed. So then B positive is only missing A antigen, so they would have antibodies against A. AB negative is only missing that RH antigen, so they would be RH if exposed. AB positive is not missing any. It has all of the antigens, so they would not make any antibodies against any other blood type, which makes them unique. So this individual would be called a universal recipient. And that's because they don't make antibodies against any blood type. So you can give them any of these other kinds of blood and they're not going to have any problem with it. Their antibodies are not going to attack it. Okay? All right. So O negative, we are missing all of the antibodies, or all of the antigens. All right, so they would have antibodies against A, against B, and against RH if they're exposed to it. Whoops. Okay. And then lastly, we have O positive. So it's only missing the A and the B, so it would have antibodies against A and B. All right, so that's our chart. Okay. So again, practice it, all right? Try just writing down all the blood types. Make your own blank sheet of paper. Don't look at your notes. Write down all the blood types and then the antigens and the antibodies for them, all right? And so just practice it. So now we're going to go and see what reactions we could have with a transfusion, all right? So I've got some kind of examples here. So we'll determine if these matches are compatible. Okay, and so the important thing to remember with these transfusions is that the donor, okay, so this is the person giving their blood, the donor's antigens, all right, so their antigens on the surface of the red blood cells cannot interact with the antibodies in the recipient's blood. Okay, so it's going to refer back to that chart that we just made. Okay, so the way I would think about this, especially when you're starting out, eventually it'll become kind of natural and you'll understand it, all right, is to kind of fill out for each. So what are the donor's antigens? All right, so if we're starting A negative for our donor, the antigens on this cell would be A. All right, and so it's negative, so there's not an RH on there. The individual with AB positive blood, if we look back up at our chart, all right, that's this individual here. What antibodies do they make? All right, they don't make any antibodies. Okay, so this person has no antibodies in their blood. All right, so none. Okay, so would the antibodies for the recipient attack the antigens on the donor's red blood cells? No, they would not. So this would be a compatible transfusion. So yes, this is compatible. Okay? All right, so what I would recommend you do is pause the video for a second and try doing these next few, all right? And then I'm going to go through them and the answers, and you can kind of check your answers. All right, so pause it now and go through them. Okay, so let's do the answers. All right, so again, we have A negative as our donor, so they just have an A antigen. The antibodies made by a B negative individual would be antibodies against antibodies against A and RH if exposed. Okay, so is this compatible? No, it is absolutely not compatible because this A antibody will attack this A antigen and cause these blood cells to clump. And so red blood cells clumping is a transfusion reaction. Very bad. You do not want that. All right, so this is not a compatible match. Okay, AB positive. So with AB positive, they're going to have all three on the surface, right? So A, B, and RH. All right, an individual with B positive blood would only have antibodies against A, all right? And so then this A would absolutely, all right, this A antibody would absolutely attack the A antigen on this donor's red blood cell. So this would not be a compatible match, so no. All right, so O negative has no antigens on their surface, all right? So none on there. We can kind of just make a note up here, so none. And then the antibodies made by an individual with AB negative would be RH if exposed. Okay? All right, and so this would be compatible, right? Because this RH, there's going to be no interaction because this person doesn't have any antigens. Okay, so one note here, just like our AB positive was a universal recipient, O negative is a universal donor. Right, and you can see that now because you can see they don't have any antigens on their surface. So you could give that blood to anyone and there's no way they'd have antibodies that could attack it, right? So they're a universal donor. Okay, and then one other point, so this is, yes, it is compatible. One other point to make, all right, with this, so this individual only has RH if exposed. However, You absolutely, in a clinical setting, never ever, all right, never give positive blood to a negative patient. Okay? All right, so that's just a rule in hospitals. Never. Even if you know without a doubt they have never been exposed, so there's no way they're going to have a transfusion reaction, never, ever give positive blood to a negative patient, because you do not want to be the reason that that patient now also they've been exposed to RH, so they now also make RH antibodies, right? You never ever want that to happen, especially we're going to find out when we talk about some problems with blood typing and pregnancies, right? So especially with women, you don't want to expose a woman to this RH antigen, causing them to make antibodies, because that could essentially give them serious problems when they're trying to have children. Okay, so never, never, never give positive blood to a negative patient. All right, so let's do these last few. Okay, so this individual would have B and RH. All right, this recipient is our universal recipient. They don't have any antibodies, none, so they can receive any kind of blood. So this is yes is always going to be the answer when the recipient is AB positive. Just like yes is always the answer when the donor is O negative. All right, so then AB positive, we have A, B, and RH. And then our recipient is going to have antibodies against B and RH if exposed. Okay? Whoops. And this would not be compatible. Those B antibodies here, right? They'll attack this B antigen. And again, we never want to give positive blood to a negative patient, okay? All right, and our last one is just going to have an RH on it. And our individual, our recipient, is going to have a negative blood, so they'll have B antibodies, and they will have RH. Okay, so hopefully you said, no, this is not compatible, because you never give RH, you never give positive blood to a negative patient, even though, right there, this B antibody is not going to interact with any of the antigens. You never give positive blood to a negative patient. Okay, good. So that's transfusions. So again, practice them, and I've got a few on your worksheet, your homework sheet, for you to try. Okay, and you'll get more and more comfortable the more times you do it. All right, so let's talk about RH incompatibility, okay? And so this has to do with a mother and the child she is carrying, okay? So this refers to when a mother has negative blood and the baby has positive blood. All right, so this can be a very serious problem. So we know that this mother, she has negative blood, so she is going to have RH antibodies if she is exposed to positive blood, okay? And so what happens with this scenario, a mother with negative blood, is that her first pregnancy, if the baby has positive blood, will be fine, right? The baby will be delivered normally, okay? The first pregnancy, baby is delivered normally, but now during delivery, what happens is some of the baby's blood will typically get into the mother's bloodstream during delivery. All right, now she has been exposed to that positive blood, so now she makes antibodies against RH, Okay, so after that first pregnancy, she will have antibodies against Rh. All right, because during delivery, the blood comes into contact from the baby with the mother's blood, so now she's exposed. All right, and so what happens with, so let's just put that here. All right, so first pregnancy, normal. All right, but the mother is exposed to that positive blood. All right, during delivery. Okay, so then the problem is going to come in the subsequent pregnancies, all right? And so with those, the pregnancies end up being miscarriages and the babies are not viable, all right? And that is called erythroblastosis fetalis, okay? So with that, following pregnancies are miscarriages. Okay, and so prior to understanding the cause for this, there would just be families who would have a child without any problem, and then after that, they would just always have miscarriages and not understand why, until we were able to figure out that it was this Rh incompatibility. And so once we had that, we were able to come up with a solution, which is RhoGAM. Okay, and so now some of you may know this from personal experience or working in the hospital, but now RhoGAM, if a mother has negative blood, the baby has positive blood, the mother is given an injection prior to delivery. All right, so this is an injection prior to delivery. All right, and so what the injection contains is antibodies against Rh. All right, so they're going to contain antibodies against Rh. And the reason that this works is you can give the mother this injection right before delivery, and then if some of the baby's blood that is positive gets into the mother's bloodstreams, these antibodies you've injected will take care of it, and it won't trigger the immune system to start making its own antibodies, right? So it's kind of like these antibodies you injected, just take over, get rid of that blood, right? That foreign blood, and there's no problem, okay? All right, and so the reason this is a big problem and this Rh incompatibility has to do with the fact that these Rh antibodies can cross the placenta. And so if a mother is making, after being exposed that first time, she's making these antibodies, they can go across the placenta and harm the baby and cause the miscarriage. However, A and B antibodies against, the A and B antigens cannot cross the placenta, so there's no interaction there. Okay, so that's Rh incompatibility. All right, so now let's move on and talk about white blood cells, so leukocytes. Okay, so we talked about hematopoiesis for red blood cells, so now let's talk about it for white blood cells. Okay, so we said before, we start with a hematopoietic stem cell, all right, or a hemocytoblast. That is a cell that can become any type of blood cell. Okay, so this initial hematopoietic stem cell can become two different types of cells. So it can become a lymphoid stem cell, or it can become a myeloid stem cell. All right, so the lymphoid stem cell will then give rise to lymphocytes. Okay, and so we'll talk about lymphocytes. There's a few different types of lymphocytes. we'll go into when we talk about the types of white blood cells. All right, but lymphoid stem cells become lymphocytes. Myeloid stem cells essentially become everything else. So they can become, remember, our reticulocytes that are the precursor for red blood cells. They can become platelets, which we'll talk about kind of the in-between cell for there. They can become band cells, which are immature neutrophils. So they're called band cells just because their nucleus is very like straight and elongated, so it looks like a band. So band cells. They can become eosinophils, basophils, and monocytes. Okay, so just remember, lymphoid stem cell becomes lymphocytes. Everything else comes from a myeloid stem cell. Okay, all right. So now let's talk some general characteristics of white blood cells. So in general, white blood cells, kind of when we're comparing them to red blood cells, they have a nucleus. All right, so they are nucleated. Okay, and they also have organelles like mitochondria and that kind of thing, all the normal organelles. All right, so that also means that they can live a lot longer. All right, so red blood cells are destroyed and old and not effective by about 120 days. However, these white blood cells can last, a very, very long time and be with you for a very long time, most of your life. All right, okay. They are also 2 to 3 times larger in size than a red blood cell. However, they're a very small fraction of the blood volume, so less than 1% of blood is made up of white blood cells. All right. Okay. So a couple terms. So white blood cells are unique compared to red blood cells because they do have the ability to leave blood vessels, and obviously that's important for their function. If they're going to the site of some sort of infection, they need to be able to get out of the blood and go there. All right, so a couple terms relating to that is first chemotaxis. All right, so chemotaxis is when a cell moves towards a chemical signal. All right, so chemo meaning the chemical signal, and taxis, think like taxiing, so it's moving towards that signal. Okay, so what happens with white blood cells is, let's say you've got some sort of injury. All right, so you have maybe a cut, and those damaged cells are going to release chemicals that are going to signal these white blood cells something's going on, you need to get over here. All right, so that is chemotaxis. So they're going to move towards that chemical signal. Okay, so in order to get out of the bloodstream, all right, they need to undergo what's called diapedesis. All right, so this is where they squeeze, and that's what the picture is showing, through the epithelial cells. All right, so these would be the epithelial cells of the blood vessels. Okay, so this is how they leave the blood vessel. So if there is some sort of like chemical signal out here, right, that they're trying to move towards, so chemotaxis, the cell is going to kind of be activated and it's going to want to move towards that. So it's going to approach that barrier, right, the edge of that blood vessel, and it is going to squeeze between the junctions of those epithelial cells and get out of the blood vessel. All right, so that's diapedesis. Okay, and so then a couple other terms relating to white blood cells. So leukocytosis, this would be too many white blood cells. All right, so someone has an overproduction of white blood cells. All right, so this could happen normally when somebody is fighting some sort of an infection. White blood cells can double their numbers within the time span of about an hour. All right, so in response to some sort of signal or infection, they can, double very quickly, and you'll have a lot. All right, so maybe not too many, but an increase, elevated white blood cell levels. That would be leukocytosis. So it can indicate your immune system is fighting something. All right, and leukopenia would be too few white blood cells. All right, and this would be a concern because this could leave you immunocompromised, right? Unable to fight off an infection if you encounter one. So that would be a concern as well, and you'd want to understand the cause of that in an individual. All right, so now let's talk about the types of white blood cells. Okay, so we break white blood cells down into two different categories. So they're either granulocytes, and so that means that they have visible granules in their cytoplasm. All right, and so these granules are just going to contain different chemicals that they'll release in order to, perform their function. Like we've talked about, histamine is, released for an inflammatory response. You probably learned about that in ANP1. All right, so things like that are going to be in the granules. And so with granulocytes, the granules are visible. With agranulocytes, which we'll talk about in a moment, they still have granules. They're just not visible under a microscope like they are with these granulocytes. Okay? All right, so neutrophils. So for each of these, we're going to talk about the percent of a white blood cell differential that they're responsible for. And so what a white blood cell differential is, all right, so white blood cell differential is they take a sample of blood, they make a blood smear, and they count maybe out of 100 white blood cells, how many of those blood cells are neutrophils, how many are eosinophils, how many are basophils and monocytes and lymphocytes. Okay, and so then that gives them a differential, so a percentage of all of the white blood cells that are neutrophils or whatever you're talking about. Okay, so we'll talk about the average numbers for those. And so neutrophils are the most common, so they make up 50 to 70% on a differential. All right, so the majority of the cells you would see on a slide of blood, the white blood cells would be neutrophils. Okay, and so neutrophils have kind of their structure is they have this multi-lobed nucleus. All right, so it has one nucleus. It's just kind of squeezed into these different lobes that can sometimes look separate, but it has one nucleus. They're all connected. Okay, and so the function of a neutrophil is that it is the first response. All right, so these are the fastest white blood cells to get to the site of some sort of incident or infection or something like that. Okay, so they're the first to the site of an infection. Their granules contain what are called defensins. Okay, and so defensins poke holes. They're really cool, right? They poke holes in membranes. So if the neutrophil rolls up to the site of some sort of infection and there's bacteria there, it can release its granules, right, degranulate, and release the contents of the granules. These defensins will go to those bacteria and just kind of like stab a bunch of little holes in the bacteria and end up killing it. Okay, so they contain defensins. They also contain lysozyme. All right, and lysozyme is has antibiotic properties. It's you probably learned about it when you learned about the eye because it's in tears. It's in a lot of body secretions, and it contains antioxidants. All right, so antioxidants have antibacterial properties. All right, so an example of an antioxidant would be bleach, right, or hydrogen peroxide. Those are antioxidants. Okay. All right. Okay, so that's neutrophils. So they're the first to the site of an infection or problem, and they're going to release all these kind of antibacterial, antimicrobial things from their granules. All right, so then next we have eosinophils. So eosinophils are between 2 and 4% on a white blood cell differential, and they are going to be functioning in fighting parasitic worms, all right, or parasites in general. All right, so it can be anything, like worms is just an example, but any kind of parasite, you'll see an elevation in the percent of eosinophils because they're going to be going to kind of phagocytize and get rid of that parasite. All right, so, and there'll also be increased during allergies. All right, so they're involved in when you have allergies, like right now in the summer, my allergies are terrible. All right, so your eosinophils would be very active then as well. So in lab, when you're looking at eosinophils or you're trying to identify one, the easiest way to tell them apart is their cytoplasm. So the granules in their cytoplasm, they're going to be kind of like orange to kind of red looking, depending. And so the way I always encourage my students to figure out what they're looking at is to always have another cell, like a neutrophil, because they're the most common ones, that you can compare it to nearby, right? So try to get what you think is your eosinophil next to a neutrophil, and so you can compare the color of the cytoplasm, and it's going to be much, much more orange for this eosinophil. And then also their nucleus has this characteristic kind of earmuff shape, right? It has 2 lobes, so it's bilobed. Okay, good. All right, and then the last of our granulocytes is going to be a basophil. So a basophil makes up 0 to 1% on a white blood cell differential. So this is the least common of the white blood cells. If you do a white blood cell differential, you may or may not even see a basophil. Okay, and so with the basophils, they have very, very prominent granules to the point where you probably won't even see the nucleus of the basophil. It'll just look completely purple and filled with granules. All right, and it ends up, those granules kind of push on the edge of the cytoplasm, so it ends up kind of looking bumpy from all those granules as well. So that's one way to tell it apart from one of the cells we're going to look at that can be very similar on a microscope. Okay, so with basophils, their granules contain histamine. Okay, and so when they release the contents of their granules, they release that histamine, and that is going to cause inflammation. Okay, so histamine causes inflammation. All right, and inflammation, remember, is redness, swelling, heat. You're going to increase blood flow to that area. You're going to attract other white blood cells, will be drawn to that histamine signal, okay? And so what can happen if basophils systemically release their contents, their histamine, that could lead to anaphylaxis, all right? So anaphylaxis is systemic. All right, and we'll talk later on about what anaphylactic shock looks like. All right, but this would be systemic degranulation. All right, so releasing of those granules of histamine. All right, so degranulation and histamine release. So essentially it's systemic inflammation. All right, and so you don't want systemic release of histamine because that causes your blood vessels to dilate. And so if all of those blood vessels all over your body dilate at the same time, your blood pressure is just going to drop right through the floor. And that is where anaphylactic shock comes in, and that's why it's so dangerous. Okay, so Those are basophils. They're not as common. They're very rare when you're looking at a blood slide, and so everybody in lab gets excited if they think they have a basophil. Unfortunately, they usually don't. They usually actually have a lymphocyte, which we'll talk about. Okay, so now our agranulocytes. So remember, they still have granules. They're just not visible. Okay, so no visible granules. All right, and so with the monocyte, these are the largest of the white blood cells. They're very big in size, and their nucleus typically has a kind of bean shape to it, like a little kidney bean shape to it. On a white blood cell differential, they make up 3 to 8% of all of the white blood cells. And these monocytes are responsible, it is the major phagocytic cell in the body, right? And they were responsible for all macrophages in the body. Okay, so a macrophage is essentially what happens when a monocyte leaves the bloodstream. Okay. So monocytes can leave the bloodstream and go to any number of places and become macrophages. You probably learned about a bunch of them, right? So when you learned about bones in A&P1, the macrophage that eats and dissolves bones, called an osteoclast, so that was originally a monocyte that went and left and found its home in the bone. All right. In the brain, we have those major macrophages, the glial cells, that are called microglia or microglia. All right. Those originally were monocytes, and then those monocytes left the bloodstream. And then we have Langerhans cells. All right, those were the macrophages in the skin. So any macrophages, and we'll come across more in the other systems we talk about, they all emerged from monocytes. Okay, so very cool. So monocytes are critically important to the function in the body. All right, and then our last type of white blood cell is going to be a lymphocyte. All right, and so lymphocytes, the reason they can be confused with basophils is because they have this giant nucleus, and sometimes this nucleus can basically just take up the entire cell, and there'll just be a little tiny sliver of cytoplasm left. And so it'll look a lot like a basophil. Okay, so these lymphocytes make up 20 to 30% on a white blood cell differential. Okay, so they're relatively common. They're just underneath the neutrophils for kind of second most common. All right, and so there are a few different types, as you can see here, of lymphocytes that we'll talk about. So they can either be very specific for a specific pathogen, or they can be non-specific where they just attack anything that's foreign to the body. Okay, so let's talk about that. So these lymphocytes are high, they're elevated during viral infections. So they're very skilled at fighting viruses. Okay, so you've probably heard of T and B cells. So let's talk about those. So we have specific immunity and we have non-specific immunity. With specific, there's two types. Okay, so there's cellular immunity. Okay, so cellular immunity is mediated by T cells. Okay, so T cells are a type of lymphocyte that will destroy cells that are infected with a specific virus. All right, so with a specific virus. Right, because remember, this is specific immunity, and so these T cells are going to be looking for, let's say we're talking about Let's say we're talking about COVID-19, right? So you would have T cells that are looking for body cells that are infected by COVID-19, all right? They won't go after maybe a cell that's infected with like influenza or HPV. They are looking only for COVID-19 because they are specific, okay? And it's called cellular because they're going to go and destroy that entire cell, right? So essentially it's going to secrete things that will kind of poke holes like our defensins, very similar to that, in the cell and kill them. Okay, and so we'll talk about this in much more detail when we do immunity. So this is just an overview. Okay, so each type of viral infection that you've encountered, and so you've made like an immune response against, you'll have T cells kind of floating around in your body waiting and looking for those specific infections. All right, so then you could also have a humoral response. So humoral refers to antibodies. Okay, so these are B cells. So B cells are a type of lymphocyte that, when activated, grow into these giant plasma cells. All right, and then those plasma cells will secrete antibodies. And so remember, again, this is specific. So those antibodies will be very specific for some sort of pathogen or virus or something like that. And we talked about how the antibodies work at agglutinating and clumping things. Okay, so they'd be made by the B cells. All right, so then for nonspecific immunity, we have natural killer cells, which I think are like, that's the coolest name you could ever give a cell, right? It's abbreviated NK cell, but I always just like to say natural killer cells because it also tells you what it does. So natural killer cells are not specific. They're like the little ninja cells of the immune system. They will kill any abnormal cell. Doesn't matter what it's infected with, all right? It could even be like a cancer cell. All right, if it's abnormal to the body, this NK cell will go and it will attack it and it will kill it. Okay, so those are our types of white blood cells. Our last topic on this video, we're going to talk about leukemia, okay? And so a couple types of leukemia. All right, so leukemia by definition is overproduction of abnormal white blood cells, and that's going to be very important. All right, so these are abnormal, not functional white blood cells. All right. Actually, we'll say abnormal blood cells because it can affect the other cells as well. It affects the bone marrow, which we learned those hematopoietic stem cells can become any cell, okay? And so pancytopenia refers to a decreased production of all cells. All right, so decreased production of all cells. Okay, so individuals with this issue, leukemia resulting in pancytopenia, so they wouldn't make enough red blood cells. All right, so they would not oxygenate their tissues very thoroughly. This could be seen with anemia, right? So they would be anemic. They wouldn't make enough white blood cells, so they would be immunocompromised, right? So they would be prone to infections, right? They would be very susceptible to getting severe infections because they don't have those white blood cells there to fight off the infections. And also they wouldn't make enough platelets. Okay, so then they would have issues with bleeding, right? So they get cut and it wouldn't clot properly. Okay, so that results from that pancytopenia. Okay, and so it can be, there are acute and chronic forms of leukemia. So acute means that it advances very quickly, All right, and so this would generally emerge from a problem with a stem cell, or it can be chronic, which is much slower and results from problems in cells later down the line from the stem cells. Okay, and so then they can also be, you can have lymphoblastic leukemia or myeloid leukemia, and that just refers to the issue, right, with the abnormal production of blood cells. There's an issue with that lymphoid stem cell for lymphoblastic. All right, so lymphoid stem cell. And myeloid, there would be an issue with that myeloid stem cell. And so then you know what emerges from each of those. So with lymphoblastic and that lymphoid stem cells, that means your lymphocytes would be compromised, okay? So then all of these cells we just talked about, the T cells, B cells, NK cells, those would be compromised. Versus myeloid, you would have problems with your red blood cells, your platelets, all of those other white blood cells we talked about, okay? So those are the different types of leukemia. All right, so that's where we're going to end this lecture, and we'll pick up and talk about platelets on the next lecture and blood clotting and the process of blood clotting.